व्हेरी गुड मॉर्निंग गाईज. आज वेळेवर आले सगळे, कसं काय? So let's start. Very good morning and welcome to Unacademy. This is Abhijit and today we are going to discuss about the excretion and osmoregulation. Okay. So before starting the lecture, it is very necessary to introduce myself. So this is Abhijit Madkar and I have more than 6 years of experience. I also cracked NET CSIR in December 2016. Okay, so today we are going to discuss about the excretion and osmoregulation on this channel of Crack Maharashtra CET. If you do not subscribe this channel on YouTube, please subscribe it. Okay, so you are here on the India's largest platform where crores of people are live on this platform per day. Okay, so thousands of educators are there and crores of students are online for different type of lectures. Okay, so if you want to be a part of an academy, it is necessary to install this app, okay, on your mobile. So to install this app is available on App Store also. Okay, so please install this app and sign in to enjoy the different type of lecture and with the different educators also. Okay, so it is also necessary to install Telegram on your mobile. So if you do not have Telegram, first install it. Okay, then after installation, add a URL which is here. Okay, and when you add this URL here or the link here, you will have a join option or the bottom of the screen and then join it. So in this channel, you will have a daily update about lectures. Okay, so time of lecture, subject, topic, and educator also. So this type of update are available on this Telegram app. So if you do not have it, please install and join us. Okay, so it is difficult to send you the link personally. So it is necessary to join the channel. You will have the different type of lectures here on the same channel. If you do not have any subscription, so it is necessary, guys. If you want to be enjoy this app completely or the Unacademy completely, it is necessary to take the subscription. If you are in 11th, I will recommend you the subscription of 24 month. The fees for the subscription is 24,000, and if you use this magic code ABHIJIT in a capital, so fees will be only 21,600 for 24 month, and for 12 month the fees will be reduced to the 14,850 with this magic code if you are in a 12th. Okay, so if you already complete your 12th, so you can take the subscription of 1 month in only 1499 rupees with this magic code. If you use this code at the time of subscription, the fee will be reduced. Okay, so if you take a subscription, there are different facilities available for subscription. So these facilities is for offline lectures you can download and see whenever you want. So there is different type of educator, so the option is there also a favorite educator, so you can interact with your favorite educator and with this you will have the doubt solving session. So special doubt solving session is also there. So you will have interaction with your favorite educator. So this type of different facilities are available with the subscription. Okay, so let's start with excretion and osmoregulation, my dear friends. Okay, so what is meant by excretion and what is meant by osmoregulation? So excretion means excrete the waste material. Okay, so it is excretion or elimination of metabolic waste. Okay, it's a elimination of metabolic waste. And osmoregulation means a balancing a salt, or you can say ions or minerals also. Okay, so this is a definition of your excretion and osmoregulation. Okay, so guys, are you there? So let's see, guys, why this excretion and osmoregulation is a necessary. Okay, so uh, in our body, in the process of metabolism, there is different type of chemical which is the move to the metabolism. So for example, there is a glucose. If glucose is there, it will definitely convert itself into CO2 plus EH2O. Okay, and the byproduct is energy, which is the main target of body. Okay, so energy is here. If you add lipid, so the lipids also oxidize in a same pattern. Okay, so energy and CO2. But if you add, or if your body is using a nucleic acid and a protein, that is amino acid. So there is a two thing. Okay, one is a energy, so you will have energy, but there is a nitrogen waste. Okay, so nitrogen waste is also there. So this nitrogen waste is always in the form of ammonia. Okay, and it is necessary to eliminate this one. Elimination is necessary. It is important because it's a toxic in a nature. It's a toxic in a nature, and that's why it is a necessary to eliminate this. So our this system of excretion is all about the elimination of nitrogen waste. Okay, so the excretory system is mainly concerned with the nitrogen waste, or you can say elimination of nitrogen waste. Excess carbohydrate and lipid can store inside of the body, but excess amino acid are not stored inside of the body. Okay, so excess amino acid, then excess nucleic acid, they are also not stored inside of the body. So that time what happen when we have excess of amino acid and nucleic acid? Okay, that time we just eliminate nitrogen from the um, amino acid or the nucleic acid, and we have a carbon skeleton. This carbon skeleton we convert it into the glucose, or you can say sugar, and the remaining nitrogen is eliminated as a waste. Okay, so it is always in a form of ammonia, and this process are happen inside of the what we call it, a liver. Okay, if there is a liver in animal, if you see lower organism, they do not have liver. Okay, so that time there are special organs, special cells are there which are involved in this type of conversion. Okay, so guys, let's see what happen if there is excess of amino acid and excess of nucleic acid. which store the carbon skeleton, and the amino or the ammonia group or the nitrogen containing products are intentionally eliminated out of the body, and that time they are eliminated through the system, and we call it excretory system. Okay, so the carbon skeleton of amino acid are used as an energy purpose, and the NH group is released as ammonia, and it excrete out by the system. We call it excretory system. Okay, ammonia is a toxic and can be converted into urea or unique acid or guanine, depending upon the availability of amount of water to excrete, and also depending upon the organism or animal which is going to be convert this ammonia into the urea or uric acid or guanine. Okay, but the most important point here is the availability of water, okay, to be excrete. So this availability of water is a key to decide the excretory product. Okay, so for example, the um, animals which are living in a desert, so they have a minimum water available. Animal which are living in a ocean, they have a lot of water, but it's a salty water. So depending upon the organism, is it feasible for organism or not to drink uh, salty water. Okay, if organism is living in a fresh water, that means there is a lot of water for organism which is available, and organism can take it as a water, and that time uh, the product of excretion will be different from the ocean organism and from the organism which are living in a desert. So let's see how they are decide the which type of product is going to be uh, what we call it excrete by the organism. Okay, so nitrogenous waste are toxic. Okay, so let's see guys how they are toxic and why there is a toxicity. If they are the part of our excretion or the sorry the part of our metabolism, still why they are toxic. Okay, so ammonia is a basic in a nature. Okay, so what is true for ammonia, it is a basic in a nature. Okay, so guys, let's see what is meant by basic. So I'm talking about the alkalinity. Okay, so what is the question, guys? Is there any? If there is any question, please ask me. Okay, otherwise I will move forward, and you do not have the complete knowledge about the topic. Okay, so this is not right. Okay, so is there anything, guys? Please ask me if you do not get the things properly. Okay, so let's see. Ammonia is a basic in a nature. That means I am talking about the alkaline nature. Okay, so pH is pH of ammonia is more than 7. Okay, so it causes change in a pH of body fluid. Body fluid means your fluid which is inside of a tissue, or you can say a blood. Okay, so the pH of blood will be increased. So enzyme get disturbed due to the change in a pH. We know that enzymes are synthesized, or the blood enzymes mostly, or the tissue enzymes which are living in the tissue. So these enzymes uh, are synthesized, or they made to be work in the 7 pH. Okay, so we call it optimum pH. Optimum pH for enzyme is 7, or the physiological pH. Okay, so physiological pH. Okay, so on this pH, they use at or they work as the maximum rate. So okay, maximum rate of reaction. Okay, so if there is a change, any change in a pH value. Okay, so what happens if the pH of a blood it increases, it will definitely slow down the reaction. Okay, it will slow down the reaction, because we know that a factor affecting the enzyme activity we already discussed. So the enzyme activity is like this. Okay, so if you see this peak is always at the 7 pH. Okay, so it's a pH value, it's a 0 here and it's a 14 here. Okay, so at this peak we call it optimum pH. And if you shift this pH toward the 14 or alkaline, so the value should be changed, or the reaction should be slow down. Okay, so change in integrity of plasma membrane. Also we know that the plasma membrane is made up of the lipid and a protein like this, guys. Okay, so what happen if there is any changes in the pH in a cell or the cytoplasm or the fluid which is moving inside of the body, that time what happen, this integrity of plasma membrane also hampered, and due to that the transport of a molecule or the salt or the minerals get changed, and there is a low integrity between the cell and the environment. Okay, so the integrity also lost uh, because change in a plasma membrane. So it need a large quantity of water to dilute and reduce the toxicity. Okay, so who need it? Ammonia or nitrogen waste. Okay, nitrogen waste required large quantity of water to dilute and reduce its toxicity. Okay, so because of that what happened, we can't store it inside of the body is difficult. Why? Because it required a large quantity of water to dissolve or to dilute, and it reduces its toxicity. They are dissolvable in water, so it is not a matter, but they require more water for the dilution and to reduce the toxicity. So okay guys, keep in mind that these nitrogenous waste products are toxic in nature, and they will hamper everything inside of your body if they encounter with your tissue fluid or the enzymes or the different thing of our body. So physiological values of our body will be changed because of this nitrogenous waste. So we can't store this waste inside of the body for a longer time. Okay, so the first product which is get from the nucleic acid or the amino acid is always ammonia. Okay, so we can classify the organism on the basis of the excretory product, if they are using ammonia as a waste product, or they are excreting ammonia as a waste product, or urea or uric acid or guanine. Okay, so we are going to uh, classify organism into this four group now. Okay, so first organism, ammonotelic organism. So this organism use ammonia as a excretory product, and the process we call it uh, ammonotelism. Ammonotelism is a process, okay, process to secrete the ammonia as a waste product. Okay, so the 1 gram ammonia required 300 to 500 ml of water. Okay, so 500 ml of water per gram of ammonia. So it is a costly, my dear friends. So we know that this much water is not available for us. Okay, because we remove lots of ammonia uh, per day from our body, and if you use this 500 ml for 1 gram of ammonia, so we required more than 20-30 liter of water per day. Okay, and we know that we hardly take a 4 to 5 liter of water per day. Okay, hardly, I use word hardly, we take 4 to 5 liter of water per day. Okay, so that means we are not made to uh, excrete the ammonia as a byproduct, or ammonia as a waste product. Okay, so the organism, who are the organism or the organism which are made to excrete this type of a product. Okay, so let's see, there are aquatic animal, aquatic invertebrates. Okay, so this is obvious, my dear friend, because this much water is only available for the uh, organism which are living in a water. Okay, so water living organism जे आहेत तर त्यांच्यासाठीच एवढं पाणी अवेलेबल आहे. Otherwise you and me can't afford this much water, or we can't take this much of water and use ammonia as an excretory product. Okay, so bony fishes are there. A larva of the amphibians are also uh, use ammonia as a excretory product. Okay, so the organs, let's see the organ which are involved in the excretion of the ammonia, the skin or the gills and the kidney. Okay, and sometimes these three organs are also involved in the um, excretion of ammonia. Okay, so let's see the example of frog. When it is in a first stage, it uses skin. Then the second or third stage, the gills are fully developed, and that time frog use gills. And then when it convert itself into the complete adult form, that time it just remove itself, or the came out of the water, and that time it can use a kidney to secrete this type of material. Okay, so the organ which are involved in the excretion of the ammonia are the skin, gills and kidney, depending upon the organism, or sometimes organism in a different stage can use all three organ as a excretory organ. Okay, so let's see the second group. So we will have already we discussed the first group, we call it ammonotelism. Okay, or the ammonotelic organism. The second group we will have is a ureotelic organisms. Okay, so this process of excreting urea as a nitrogen waste, so this process is called as ureotelism. Okay, so the process is called as ureotelism, guys. Okay, लक्षात येते का रे, जे अमोनिया बाहेर टाकतात त्यांना आपण म्हणतो अमोनोटेलिझम, बरोबर. आणि सॉरी, अमोनिया बाहेर टाकण्याच्या प्रोसेसला आपण म्हणतो अमोनोटेलिझम. आणि जर ऑर्गॅनिझम अमोनिया ऍज अ नायट्रोजन वेस्ट बाहेर टाकत असेल तर त्या ऑर्गॅनिझमला आपण म्हणतो अमोनोटेलिक ऑर्गॅनिझम. ओके, आता आपण युरिया बद्दल पाहणार आहोत. युरिया जर बाहेर टाकीत असेल तर त्या प्रोसेसला म्हणायचं युरिओटेलिझम, आणि ऑर्गॅनिझमला म्हणायचं ureotelic organism. Okay, so this is your second group of organism which is using urea as a byproduct. Okay, so let's see guys, when 1 gram of urea is excreted, it required only 50 ml of water, and this much water for 1 gram of ammonia is available for us also, so we can be a ureotelic. Okay, so let's see we are ureotelic or not. Okay, so the terrestrial animal. Okay, so the one animal which are living on the earth or the surface of earth. Okay, so we can call them a terrestrial animal. And the second group of animal we generally use term the aquatic animal, so they are living in a water. So this terrestrial means they are living in a, sorry, living on a earth or the surface of earth or the soil, in a soil. Okay, so mammals, so we are the mammals, we are from this group also. Okay, so guys, we are also using urea as a waste material. So cartilaginous fish. Okay, so guys, do not get confused. I am right here bony fish in the ammonotelic organism and cartilaginous fish in the urotelic organism. Okay, so cartilaginous fish means this organism or these species are also called boneless fish. Okay, so these are the boneless fish. Now we know that when we classify the organism or the fish, we classify into the two group, cartilaginous and the bony fish. So bony fish are ammonotelic and the cartilaginous fish are urotelic. Okay, so aquatic reptiles, reptile organism which are living in a water, so they are also a ureotelic. And amphibians, so most of the amphibians, not all of the amphibians, but most of the amphibians are ureotelic organism. Other the remaining amphibians are use the ammonia as a excretory product. Okay, so let's see what happen when we know that the nitrogen waste is always ammonia. So body use or body prepared making ammonia from nitrogen or the direct ammonia is a nitrogenous waste. Okay, but when organism want to convert it into a urea, or so organism want to excrete a urea, that time it is necessary to convert this ammonia into a urea. Okay, and this process required uh, a set of enzyme, and this set of enzyme is in the urea cycle, or we simply call it ornithine cycle. Okay, and this cycle is always run in the liver of organism. That's why the organism you are taking as example, each and every organism contains its own liver, okay, inside of the body. So these are the liver containing organism which can use uh, a liver or ornithine cycle to convert ammonia into urea, and then urea uh, drop into the blood, and then blood separate it from the blood, and then urea is excreted by the kidney. Okay, so the conversion of ammonia to urea required 3 ATP. Okay, so guys, this is a energy consuming process. It required a 3 ATP. ADP plus PI. Okay, so this is energy consuming process. So the organism which want to conserve the water. Okay, so the conservation water required energy. Okay, so guys, it doesn't mean that the storing or the storage of water inside of our body required energy. Okay, so I'm not writing there in this sense. Okay, so what is the meaning of that sentence? Actually, the conservation of water means, guys, let's say for example, organism is going to eliminate ammonia as a waste. That time it required 500 ml per mg. Okay, so this time this much water is not available. For example, it is available, but organism required to conserve the water, so that time organism use urea as a byproduct, and only 50 ml of water is excreted with the urea, but it required a 3 ATP to convert this ammonia into a urea. Okay, so the 450 ml, we can say water is conserved per gram of ammonia to urea conversion, but it required 3 ATP for the conversion of ammonia to urea. Okay, so this is how organism whenever want to conserve the water, so there is a requirement of energy to convert this product which is taking more water to the product which is less toxic, or which is taking less water to eliminate. Okay, so that's why I use it there. The conservation of water always required energy. So the uh, meaning of this sentence is a different, but you can write it like this. Okay, so let's see the third group of organism which is not going to excrete ammonia or urea. Okay, but it convert ammonia directly into the uric acid. Okay, and uric acid is less toxic. Okay, ammonia is more toxic. Urea is less toxic than the ammonia. Okay, and uric acid is much more less toxic than the ammonia, and less toxic to the urea. So it required only 5 ml of water per gram of uric acid. Okay, so now organism can conserve more water, can be conserved by organism, but it required energy to convert this ammonia into uric acid. So there is a required of energy, but water is conserved inside of the body of organism. So let's see, guys, what happened. Now here the organism is going to excrete the uric acid as a nitrogenous waste. Okay, and it required only 5 to 10 ml of water per gram, or sorry, per milligram of sorry, I write here a gram. Okay, so it's a mg, my dear friends, per milligram of uric acid. Okay, so let's see what happened in inorganic pathway. In a liver of a bird, ammonia is directly converted into the uric acid. So ammonia is going to convert into a uric acid, and the pathway or the set of enzyme which is required to conversion of this ammonia to uric acid, we call it inorganic pathway. Okay, and it was present or it is present in the liver of a bird. Okay, so in a liver of a bird, there is inorganic pathway which can convert ammonia to uric acid. Okay, so guys, let's discuss once again. Actually, what happened? Ammonia is secreted by an organism which are with a more water. Okay, so more water is available that time, organism is going to secrete ammonia. If water is less, then organism is going to secrete urea for the less water. Organism which is in the area having a less water. Okay, when water is uric acid, water is a very less in amount which is available for organism that time, the bird or insect, a very low water is available for this group. Okay, so very low water is available for this group of a bird and an insect, and that's why they prepare the uric acid as a product of excretion. Okay, so Mansi, actually this process I do not have total detail of this Seneca pathway. Okay, or I can't give you because this is not a part of your syllabus. Okay, so we can't discuss this Seneca pathway in a very detail. Okay, in your biochemistry, you will have only the pathway like a glycolysis, Krebs cycle, uh, then C3, C4, which is present in a plant, and then electron transport system. Okay, so we do not have any pathway like inosinic and uric acid. The Cori cycle is not part of our syllabus, so we never discuss it. Otherwise, Cori cycle is a very good to discuss, and the uric acid cycle is or the urea cycle is also very good to discuss in under this topic. But it is not a part of our syllabus, so we are not going to discuss it. Okay, so I'm extremely sorry I cannot discuss or the cannot give you a detail about this scenic pathway completely. Okay, so but we can find the ATP which is required to conversion of ammonia to uric acid. Okay, and we will definitely discuss in the next lecture when we discuss about the we have a total 3 lectures on this excretion topic. Okay, so today we are going to discuss only these basic things which are related to the uh, excretion. Okay, today we will see we will discuss the physiology or the complete structure of human kidney and the functional and structural unit of human kidney, and at the lecture in the third lecture, we will discuss the control on the kidney or the excretion of the product. Okay, so very good, and thank you very much. Okay, let's see, guys. Uh, it required a 5 to 10 ml water only to conversion or the to dissolve 1 mg of uric acid and excrete out. Okay, so enosinic pathway is a pathway which is in the liver of a bird, which is conducted in the liver of a bird, which convert ammonia to direct into uric acid. Okay, so the organism which having a low supply of water is a bird, insect, reptiles, and a snail. Okay, so these are the organism which required very low amount of water to excrete the product of your nitrogen. Okay, so ammonia or the nitrogenous waste. Okay, so we have guys one more question. What makes urine pale yellow? Okay, so yes, this is part of your syllabus and I can explain definitely. So it is, I also uh, write this point in your second lecture, but no issue. So there is a Eurochrome, which is yellow in a color. Okay, so it is a result in the pale yellow color of urine. Okay, so it is synthesized in a liver also. Okay, we know that a liver had a Kupper cell. Kupper cell. Okay, so the Kupper cell in a liver are involved in the destruction of RBC. Okay, or killing of RBC. Destruction of RBC. Why? Because the age of RBC is only 120 days, and after 120 days, or there is any damage to the RBC, or the RBC is a block by a carbon monoxide. So there are different type of things which can comes between in the function of RBC. That time it is necessary to kill this RBC and make a new one, because RBC is a dead. We know that this is a dead. So this dead material inside of our body never start repair it. Okay, so body never repair it, just replace it. So now here is also necessary to replace this RBC. So before replace, it is necessary to kill it, and this RBC is killed by your Kupper cell, and that time there is a two product from RBC. One is a protein part, and the second is a ferrous. Okay, so iron part. So this iron part is a store inside of the liver with the protein. We call it ferritin. Okay, so ferritin protein helps to conserve or this storage of ferrous. Now we have a protein part. This protein part can be converted into three groups, or we can say here is a urochrome. Okay, so urochrome is also a part of destruction of your RBC, and there is also two products which is a part of destruction of RBC. One is a bilirubin and one is a biliverdin. Okay, so these two products are excreted in the digestive system. So the feces or the undigested food. Okay, so yellow color of undigested food. We call it a feces. Okay, so the yellow color of undigested food is due to the bilirubin and biliverdin, and the yellow color of your urine is because of the urochrome. Okay, so these are the two components which is responsible for the yellow color in a digestive system and excretion system. Okay, so this yellow color undigested food, or the we can say feces. Okay, so it is not your excretory product. Okay, so excretion is uh, a different, guys. Okay, so defecation is a different. Okay, defecation is a different, and excretion is a different. I already explained in the first 5 minutes. If you do not attend the lecture in the first 5 minutes, so please after lecture you can see this lecture from the first point. Okay, so there I already explained what is meant by excretion and why a metabolic product is important to use this metabolic product word in the definition of excretion. Okay, so we have one more question. How does body heat affect our urine color? So project, actually body heat does not affect our color of urine. What happen, the when our body is heated, okay, due to the any infection, so that time the function of body is not moving like a normal, and because of that there is a more or less production of urine, or the more of or less production of this type of color component, and it will definitely affect our color of urine or the different functions. Okay, so let's see what happened, guys. There is a 37 degrees Celsius. a temperature which is optimum for our enzyme. Okay, if there is any change, it's a 36 or a 39. Okay, so it will definitely reduce the function of enzyme. So what happened, the enzyme will not work properly. Okay, not work properly, and it will result in the different type of uh, symptoms in our body. Okay, so guys, uh, your your body he do not actually directly uh, affect the function or the do not affect the color of your urine, but there is an infection which result in the heat of the body. Okay, and then it will affect the function of enzyme, and then we will have symptoms like this. Okay, so these symptoms are because of the function of enzyme. Okay, so when your body is heat, there is also affect or effect on your taste. Okay, so your taste buds also for example, this is a taste bud. Okay, so different type of taste is due to the different type of taste bud in your on your tongue. And when temperature increases, for example, temperature get increased, so taste your taste bud may be change its a shape, or we can say they are denatured, and because of that there is uh, a loss in your taste, and it results in the taking a low amount of food because of this loss in the taste of the food. Okay, so this is how the body is affected by the different thing uh, in our daily life. Okay, so let's see. So we know that this example of organism is example of organism will have a very low supply of water in their daily life, so they are using uric acid as a nitrogenous waste. Okay, so let's see now Goni. This is a fourth part of organism, and there is only few organism in this group. The one is a scorpion, a second is a spider, and the third one is a penguin. So these three organisms are in the area where a very low amount of water is available. Okay, so they excrete a guanine. Guanine is a not a toxic material, not toxic at all, because we know that guanine is a part of a DNA. Okay, so ATGC. So this G is a guanine. Okay, so this G is a guanine, and it is the organism which is containing G or guanine uh, as a product or as a waste material. That time, this guanine is not going to harm uh, into the body, and organism can store it for longer. Okay, so the actually what is the matter? Ammonia is a toxic, so there is a very less time to store it. Otherwise, it will harm your body. Okay, so it required more water. Urea is a less toxic, so body can hold it for a few hours. Okay, so it can be hold for a few hours, but it required again a 50 ml per mg. Then uric acid is a very low toxic, so it can be stored for a longer. Okay, and guanine is a very low, or it's not toxic at all, so body can store it for a longer, and that time the guanine, which is a waste of organism, this organism can secrete it in a jelly form, or you can say a solid form. So it required no water, you can say a very less water or no water, because of its pH value. Okay, so guys, why ammonia is a highly toxic? We have a question. So because of its pH value. Okay, its pH value is a more than 7. That is alkaline. It's basic in nature. It will hamper your pH. Okay, and if your pH of a body will get, it will result in the uh, a malfunctioning of enzyme. Enzyme will not function properly, and it will result in the thousands of symptoms in our body, or thousands of enzyme will get damage due to the low pH of blood or low pH of your body. Okay, so this is very necessary to maintain the pH of a body, and any chemical which is going to hamper the pH of your body, our body will get reduced it, or the remove it from our blood. Okay, so the excretion is a best way to remove that product from our body. Okay, so they eliminate the jelly like or a dry product, and this guanine is not a toxic at all. Okay, so guys, if you write it, just take a screenshot and then keep it as your part of the notes. Okay, so guys, please start making a notes from now. Okay, आतापासून नोट बनवायला सुरुवात करा. शेवटी शेवटी काही होणार नाही. 11th आरला आतापर्यंत आपल्याकडनं काही झालेलं नाही. आपल्याकडे मागच्या 10 वर्षांचा किंवा we have experience of 10th class. Okay, and we know that at the last hour we will have no any planning for the best score. Okay, so it is necessary to plan it from now. Let's see osmolarity and organism. Why there is a osmoregulation inside of our body, and why it is maintained. Okay, and why it is maintained by our body. Okay, so there are two type of organism in the nature. One is osmoconformers, and the second group of organism we call it osmoregulators. Okay, so these are the two group. We can classify the organism on the osmolarity or the maintenance of osmolarity. Okay, so the first group you can see these osmoconformers are the organism which can maintain an internal condition equal to the osmolarity of their environment. Okay, so let's see guys, for example, this is your organism, and you are putting this organism in the water. So the internal environment of this organism can be maintained with the this external environment. Okay, so if you see the salinity of external environment is 10%, and the internal environment of organism is a 5%. So organism will allow to enter this salinity inside of the organism, and it will maintain at the 10. Okay, so this is how the organism can change their salinity with respect to the environment, or the osmolarity of the environment. Okay, so this type of organism we call it osmoconformer. If you see the second group of organism, these are the organism which can able to tightly regulate the body osmolarity, and they are the osmolarity of a body is not depending upon the osmolarity of environment. Okay, so the irrespective of environment, they can maintain the osmolarity of our body. So we are organism which came from this group. We can maintain our osmolarity of our body. Okay, so this is how there are two type of organism in the nature. One can confirm one can be a osmoconformer which can change the internal condition of a body with respect to the condition of environment, and the second osmoregulators, they can regulate the osmolarity of internal environment irrespective of the environment. Okay, so body fluid and the external environment is isoosmotic in a nature. Okay, so what is the plan of this osmoconformer organism is to maintain the isoosmosity or isoosmotic condition in the internal and external environment. So what happened? So external environment will not going to apply any more osmotic pressure on the body of organism, and the internal part of the body also not going to apply any osmotic pressure on the wall of the body due to the change in concentration difference, or the due to the concentration difference uh, between internal and external environment. Okay, so this is the ultimate plan of a organism to maintain the isosmotic conditions. Okay, let's see. Minimized osmotic gradient result in a low movement of water in and out of the cell. Okay, so minimized osmotic gradient results in a low movement of water in and out of the cell. That is what I explained. So if there is a minimum difference between the external and the internal environment of organism, so that time the movement of solute uh, in and out of the body is automatically reduced because of there is a very low pressure, osmotic pressure, which is available or which is a present. Okay, so the disadvantage of this osmoconformer is the internal condition may be a suboptimal. That means the change in environmental condition which always result in the change in a internal condition of the body. Okay, so let's see guys. Most osmoconformers are the marine invertebrate. Okay, so it is obvious guys, because the change in environment or the change in osmotic pressure is hardly or it is easily happen inside of the marine organism or the marine environment. Okay, so let's see, for example, this is a sea. Okay, so there is a points where a river gate enter inside of the sea. So that time here we call it a brackish water, and this water shows high salinity, high salt concentration as compared to the river water, and low salt as compared to the uh, ocean water or the sea water. Okay, so due to that, here is a difference in a salinity. So in a river it is a different, and in ocean it is a different. So when organism maintain want to move from these three different salinity or three different type of water, that time it is necessary to maintain its salinity inside of the body. Otherwise, the organism should be a osmoconformer, sorry, osmoregulator, or it should be a osmoconformer. When it is osmoconformer, the organism or the internal environment of organism is here hyper osmotic, like water of sea. At the point here, it is subosmotic. So the salt concentration is a minimum or the smaller than the seawater, and when this organism enter into the river, that time there is a few salt present inside of the water, because there is a very few salt present inside of the uh, river water. Okay, so this is how this organism can change their optimal condition or the internal condition of the body with respect to the environmental condition or the salt concentration in the environment. Okay, so the starfish is a very good example of the osmoconformer. Okay, and bony fish is a very good example of the osmoregulator organism. This organism can tightly maintain the osmolarity irrespective of the uh, condition of environment. Okay, osmolarity of body stay always constant. So the osmolarity of body stay always constant means, for example, there is a for example 30% of a salt inside of the. Okay, so it's just example, I'm not going to target any organism here. So for example 30% salt is present in a cytoplasm. So at any cost, if there is any change in the environment or the uh, environmental condition, the cytoplasm always shows a 30% salt. Okay, so it is not going to show more than or less than 30% salt when the organism changes environment. Okay, so the osmolarity of the body fluid is always maintained, or it is always constant. Kidney plays main role in osmoregulation. We know that if there is extra salt, extra, okay, salt or ion or solute in our body fluid, kidney will remove it. Okay, so do not this word, do not use this word remove, use like excrete it. Okay, kidney will excrete it. If there is a low concentration of salt in our body, that time kidney do not excrete it, kidney reused it, and again drop it into the blood. Okay, so this is how the kidney regulate the or main plays main role in osmoregulation. Okay, so guys, are you getting my point? What is the difference between osmoconformers and osmoregulators? Okay, please be concentrate. We have another two group of organism depending upon the osmolarity, and we are going to discuss it today itself now. Okay, so if you see the disadvantage of maintaining maintenance of osmolarity is a use of ATP. Okay, so the uh, osmoregulation in a body required energy, and osmoconformer do not require energy because it's a passive process and it's an active process. Okay, it's a passive process driven by osmotic pressure, and it may be opposite to the osmotic pressure. So it required energy. Okay, and it is driven by the osmotic pressure, so do not require energy. Okay, so this is how the organism required energy to maintain the osmoregulation, or the organism do not require energy when they are changing its environment, internal environment, with respect to the external environment, because this type of changes occur or they are driven by the osmotic pressure, which is generated due to the change in a salt concentration or the difference in a salt concentration inside of the body and outside of the body. Okay, so let's see guys, what is there more there in the sense of internal environment or the salinity. Okay, or the salt concentration. Okay, organism can be again classified into a two group, stanohaline and the urohaline. Okay, so stano, the word steno means a narrow. Okay, so it is a narrow, and the urohaline is used term used for the wider range. Okay, so let's see, when organism only tolerate narrow range of salt concentration, that means for example, organism is going to hold itself as a normal in the very narrow range, or the when narrow changes in the concentration of a salt, at that time we can say stanohalines. So our the conformers. Okay, so we already explained here osmoconformers. Most of the osmoconformers, most of the osmoconformers are stanohaline. Okay, they can change its body environment, but for very narrow range. Okay, and the second group of organism is a urohaline organism capable of handling wide range of salinity. That means organism can live in 0.5% of salt to the 5% of salt. Okay, so this is a wide range, and if we are talking about the this salinity, the organism can live maximum 1.2 or 1.5. Okay, so these are the just examples, guys. So I'm not going to point out any organism here, so this organism can live. It's just example. Let's take an example 0.5 to 1. This is a narrow range, and 0.5 to 5%, it is a good range of the salinity where organism can maintain its body environment. That time you can say this organism is urohaline, which can maintain its 0.5 to the 5% of salinity, and the organism which maintain itself in the only 0.5 to 1%, that time you can say it's a stanwala, so it can tolerate only narrow range or the narrow changes in the salt concentration. Okay, so unicellular organism, let's say there is a different type also. The unicellular organisms are there which have contractile vacuoles to store and to eliminate the waste product. Okay, so in unicellular means organism which is made up of single cell, they have one vacuole type structure or the sac which store the excretory material, and it will automatically remove out, or the this vesicle will go and remove itself inside of the outside of the body, that is in environment. Okay, so this type of vesicle are present in a unicellular organism. So we know the simplest form of animal is a sponges. Okay, so sponges diffused is waste material through the osculum. We know there is a uh, pores on the body of these sponges. If you do not remember the body of sponges, let me memorize you the things. Let me draw it. Okay, so the sponges your looks like this. We already know, and in our book the diagram is something like this. Okay, and there is a pores on the organism. That's why we call it a porifera. Okay, so fera means bearing. Okay, and pora means pores. Pores means holes. Okay, so the whole containing body yeah, organism, and this organism remove its waste material from this hole. We call it a vasculum. Okay, so please revise this porifera chapter. I mean, chapter is a classification of animal. Okay, it is used for the 1 or 2 marks in examination. So it is up to you. If you want to revise it, you can revise it. Okay, so the example of this is sponges. Okay, so this organism we call it sponges. I already like write here sponges, but the group of organism in the name of a phylum we will find the porifera name. They have osculum, or the main spore by which they eliminate the excretory product. So there is a one bird, a marine bird, we call it albatross. Okay, so this bird spent years uh, on flying on the ocean. Okay, so guys, let's see. For example, a bird is inside of the ocean. We know the ocean do not have any corner. Okay, so it is a longer and larger on earth. Okay, 70 or the 72% of water is only uh, held by the single oceans also. Okay, so the when this bird is going to spend its maximum time on ocean. Okay, that time what happen, a bird do not have any water to drink. Okay, so it drink a salty water. Okay, so how can this bird can drink a salty water? So let's see, guys, what happened. Actually, this bird have a salt gland near to the nostril. Nostril म्हणजे नाक, nose. Okay, so the whole of nose, the space of a nose, we call it a nostril. So near the nostril, this bird has a salt gland. This gland can secrete a salt by active transport to maintain the osmolarity and the water iso. It made water to the isosmotic to the body fluid. Okay, so this type of modification we call it adaptations. Okay, so adaptation, this word is used in evolution. Okay, so this type of adaptations are also there with the excretory system inside of the different type of animals. Okay, albatross is a bird, and this bird have a salt gland, okay, near to the nostril. That's why it secretes. uh, okay, so guys, we have same question, how does the albatross secrete salt? Okay, so let's see, guys. For example, this is a pipe by which the water is drinked by the albatross. So here is a gland. This gland absorb the salt, and they secrete salt again out of the body, and the only the water is moving inside, which is iso. osmotic to the body fluid. Okay, this isosmotic, or you can say the isotonic. Okay, isotonic is also a good word which is used by your books. Okay, so this was the different organisms, and we can classify them, or we classify them on the basis of the excretory systems. Okay, so there are a different type of excretory system in the lower organism. So this part we are going to discuss in a very fast way. Okay, so why? Because it is just a revision of your classification of organism. Okay, so you classify this organism into the chordata, and second one is a non-chordata. Okay, so we are going to discuss here about non-chordata, most of the. Okay, so the common type of excretion in lower organism is an apradia. So there is an apradia. It is a tube like a cellular structure which is involved in the excretion of nitrogenous waste. Okay, so the first type of nephradia which is famous in nature is a protonephradia. It is a network of branch tube which is made up of a flame cell. Okay, so this is how the nephradia looks like. So this is a protonephradia, a design of a protonephradia at the edge of a organism, or the close to the near of the wall of organism. Okay, so this is how it is arranged in organism. If you zoom in this part, it will looks like a network. Okay, so this is there is also the flame cell present, or the bulb of a flame cell present. So it is complex structure made up of the branch structures. Okay, and this flame cell is going to secrete the nitrogenous waste. Okay, so here is a nucleus of a flame cell, and this part, or the cilia, will help you to remove the product which is a nitrogenous waste. Okay, so this is how protonephradia works. Then the second structure, which is again famous inside of a nature in a lower organism as an excretory product, this structure is called as a metanephradia. It is unbranched coil tube connect to the body cavity by nephrostomes. Okay, so the body fluid enters into the nephridium through the nephrostomes and get discharged by the nephrodopore. Okay, so what happened? So this tube is a nephrostome, we call it. For example, this is a tube. Okay, so it is connected to the body fluid, and this connection, or this hole, is called as nephrostome. Okay, and when it is out of a body, so this hole is called as an nephridopore. Okay, so this is called as nephridopore. Okay, so this is how this structure is arranged. Okay, so you can see this is a structure of metanephradia in an earthworm. Okay, so mostly it is present in earthworm, or the it can be a study in a very good manner in earthworm. Okay, so these are the different component of metanephradia. So if you do not read it, so not necessary to mug up, or the keep in mind this all type of thing. Okay, just keep in mind what is an apridopore, what is an apridostome, and what is an apridium, and what is a metanephridium, or these are the part of a metanephridium. Okay, so it is sufficient to um, attempt the question in examination, if any. Okay, so let's see, there is also Malpighian's tubule, which is a modified form of excretion in most of the insect, or the you can say arthropoda. Okay, so the most of the arthropoda depend on the Malpighian tubule in the process of excretion. So you can see these green color tubules uh, in the diagram are the Malpighian tubule, which are removing the nitrogenous waste from the body parts, different part of a body, and they are excrete it into the rectum anus. You can see this, that is digestive system. Okay, so the excretion is directly connected to the digestive system by your Malpighian tubule, which is actually specialized for the excretion or the absorption of nitrogen waste and secrete out into the digestive tract. Okay, so guys, please write it down. Okay, so there is Echinodermata. Okay, we know Echinodermata means what? Uh, this type of organisms, or the this is a phylum of organism which excrete a nitrogenous waste by the tube pit. Okay, so through the tube pit, I do not add this diagram here. Okay, so it is not necessary to see the diagram, but this diagram can give you idea about the Malpighian tubule, and Malpighian tubule is a hot topic as compared to the Echinodermata, excretion in Echinodermata. Okay, so the Echinodermata and the other two three organism, we know there is a system like water moving system inside of that, and this system is also help to excrete the nitrogenous waste. Okay, so that type of system we call it a water vascular system also. So it is also help in the excretion of nitrogenous waste. Okay, so let's see, in a mammal we have an apron to excrete the nitrogenous waste. Okay, so guys, uh, the apart from the mammalian excretion system to the other thing. Okay, so the neurons and the other part, we will definitely discuss it tomorrow. Okay, so we have tomorrow at 11 a.m. lecture. Okay, so on same time we will discuss the second part, where we will discuss the kidney. Okay, structure, function, and the formation of urine. Okay, and today we have a plant tissues lecture at evening at 7 p.m. Okay, so please attend this lecture. It is more important. We know that this uh, part of a plant tissue, it is not going to revise anywhere inside of your syllabus, so it is necessary to attend this lecture here. Okay, at today's evening, today evening, so be on time guys. Okay, we will going to discuss about the plant tissue at today evening, and tomorrow 11 o'clock that means a morning on the same time we will discuss the kidney and the mammalian system of excretion. Okay, so if you do not subscribe this channel, YouTube or Telegram, or I'm also talking about the uh, subscription also here, so please subscribe our channels and subscribe Unacademy to enjoy the more education which is necessary for your examination. Okay, so let's see guys, have a good time. We will definitely see you at 7 p.m. Okay, तोपर्यंत have a good time. चलो bye bye.